We welcome now move on to our next speaker, Dr. Nieforth. Welcome, Dr. Keith, to our second version of Global Virtual Healthcare Summit conducted by AKT Health Analytics in partnering with them. And Dr. Keith is a Senior Director for Certara Software, which includes Phoenix, NLME, Piranha, and Trial Simulator. Keith has more than 20 years of global drug development experience and held a variety of positions within clinical pharmacology at Roche, from direct project support role to supporting successfully for NDAs to a member of global clinical pharmacology leadership team as a system experts of the department. In addition to the expertise in quantitative and system aspects of the drug development, Keith has a passion for understanding and optimizing the human aspects of successful system implementation. Keith has a Bachelor of Science in Pharmacy, the University of Connecticut, and PharmaD from Massachusetts College of Pharmacy. Dr. Keith, you can share your screen and you can start the presentation. And I request participants, you can put your Q&A so that the end of the session can be answered.
Good morning. Can everybody hear me okay?
Yes, sir. We can hear you on. You can see the screen.
And the slides are displayed. Excellent, excellent. Well, thank you very much. It's a pleasure to join you this morning to talk about RSpeak's NLME. I think over the next 20 minutes, what I'm going to do is give an introduction to this tool. I'm going to dig into a little bit into the actual workflow because I think it's important with respect to how we've designed these tools. Talk about the pharmacometric workflow, talk about some of the challenges of implementing R individually and as an organization, and then give a brief demo of how we've designed these tools to meet some of the requirements of the pharmacometric workflow. All right, so what is RSNLME? RSpeak's NLME is a collection of open source R packages. They support pharmacometric modeling work using Certara's NLME engine and pharmacometrics modeling language, or PML, directly from the R command line. We've designed the tools with R Shiny apps that support key workflow steps, and in doing so, the tools themselves can teach the syntax of the R code. And overall, the system has been designed to provide a Certara supported environment for pharmacometric work in R. One of the things I'm going to touch on is why we believe it's important to support this type of work in R. Again, because it leads to how we've designed these tools, kind of the strategy behind it. A fundamental theme is that the tools themselves generate code for you as either a final product or a starting point for something custom. So there's complete flexibility with this approach and a concept of tools that teach. So taking a look at the pharmacometric workflow, the job, what is the job of a pharmacometrics analysis? And, many times it's discussed and kind of the focus is on the execution. However, execution of the modeling work is really only it's a significant part, but it's only one part of the overall job. And you can split the overall job into four main areas. There's a setup phase. This is where a data set is assembled from source data, which may be coming from different areas. This is compiled into an analysis data set. The data set is then explored and QC'd. Errors are corrected, and the analyst usually does graphical analysis to determine starting points for what type of model to begin with, and what kind of approach to take the overall analysis. The next phase is execution. This is where the bulk of the analysis is conducted. It's a very iterative step. Actually, the whole workflow is very iterative. It's a nature of the work. But here is where model is executed, a program puts out a lot of output, tables, listings, and then this output is post-processed. Graphics are prepared to evaluate the performance of the model, and then decisions are made around next steps. How should I adapt this model and run the next model? At the end of this process, the analysis is essentially considered finalized, or a final model is available. Then comes the reporting phase, which is where final outputs for the model are prepared, and deliverables usually include a report plus some data deliverables, perhaps some submission packages for a regulatory authority. And finally, the report phase is usually interchanged with something being done with the model, right? This is why we develop models in the first place. The models are used to solve a problem or aid a decision in the clinical development process, whether it be a clinical trial simulation, to design a better next trial, or to answer a question for an existing problem. So that's the overall workflow. Historically, users have reached for multiple tools to work their way through this workflow. Pick the best tool for the job is generally the approach. One might use one tool for data set assembly, another for data exploration. One might do the model execution and development in one software, but when it comes time to simulate, they move to another software. And this is a very efficient approach for a specific task. There's familiarity with the tool sets, task-specific efficiency. However, this approach requires frequent tool changes, and tool changes can cause significant rework and QC overhead. An example of that would be if you've developed the model in one piece of software and then you switch to another for simulation, the model needs to be recoded. Recoding the model itself is not that difficult. What's difficult and can take time is ensuring that that model is actually performing exactly the same way as it did when you developed it in the two separate softwares. So while efficient for a specific task, the cross-task efficiency can suffer with this approach. One of the most, I guess, difficult parts of using many separate tool sets across the workflow is that iterations can be extremely time consuming. And again, the nature of this work, exploratory development, it's an iterative process. You can expect to need to iterate. And a very simple example of that is an analyst gets to the point where they've drafted a version of a report. They painstakingly incorporated figures into a Word document that they've developed in another graphics package. And they send that report out for review, and they get comments back from the clinical team that perhaps, okay, we don't want to use the drug code for the legends. We want to use our newly determined market name for the report. And you can be talking hours to days to go back and regenerate in a separate tool and incorporate that. So again, a very simple example, but a very practical example of some of the deficiencies of stacking together individual tool sets for the task. So why do people reach for tools like R? Number one is a tool like R, which is a programming language, gives you a common thread across the workflow. And what that leads to is exceptional iteration efficiency. If you have your full analysis in one language in one script, you can easily go back to earlier steps, make adjustments, and rerun the code. And full analysis reproducibility is also easily achievable. Using graphical user interface tools, separate tools, it's always hard for users to go back and remember, because they're point and click menu selection. How did I exactly do that at this point in the analysis? With a command line language, it's all documented in a script and very easy to reproduce. So the issue, though, is R is a very difficult language to master, particularly for users that have not previously used a programming language. So despite the difficulty of mastering language like R, our use in pharmacometrics continues to grow. At professional meetings, it commonly comes up, the 2019 ACCP annual meeting. Many of the sessions were related to open source software in development, including R. This is from a presentation by Mark Gastonguay, kind of making the point that over time, there's a funnel-shaped expansion of open source solutions being deployed in pharmacometrics. Many of these are R. There is, gross ways to look at this. A Google Scholar search of R and pharmacometrics, looking at hits by year from 2010, shows a addition of 300 publications per year, roughly, since 2010. So a very gross way to look at it. However, the slope is going in the direction that one would expect. Professional organizations are popping up. R in Pharma, probably the most notable. This is a dedicated conference to the use of R in Pharma. It was formed in 2017, and the participant growth in that has been exponential. Looking more directly at R in pharmacometrics, we can look at CRAN package downloads. I think most are probably aware, but R is a software where users can contribute what they call packages, and these are domain specific sets of code that help users in a particular discipline do something more efficiently. So it's like a dialect within R. It's like a language, an R language within R that users can basically use to do something that they need to do for their domain. And if we just pick a sample of 6, these are 6 popular open source packages. Expose is a post-processing tool that was developed for non-mem. MRG Solve is a simulation tool from Metrum Research. We have an open source nonlinear mixed effects modeling tool, some tools for non-compartmental analysis. TidyVPC is a tool that we actually released to open source, my team, earlier in the year, and it's for a GUI environment for preparing visual predictive check diagnostics, which is a simulation diagnostic commonly used for pharmacometric models. And if we look at, these are daily downloads. So the number of downloads every day over the last 4 years, again, the slope is in the right direction. We see some variability. However, in a discipline where it's estimated there's 1,500, roughly based on professional organization membership, pharmacometricians globally, these are significant numbers, and certainly the cumulative downloads of these, again, specific pharmacometric specific R packages, the slopes are all going, all rising, quickly. There's probably other packages we could look at, like ggplot2, some of the differential solver packages, but those would not be pharmacometric specific users only. So I didn't include those in this slot. So what are some of the challenges with the adoption of R? They can be both individual and organizational. From an individual perspective, I touched on this before. Mastery of R requires a programming mindset. In my experience, there are some users who simply don't have this. Extremely talented, developers, clinical pharmacologists I work for, some of them just aren't programmers. However, with the right training, many individuals can become extremely proficient R users, but it takes some work. One of the, another challenge with R is there's many ways as a language, there's many ways to do the same thing. There's over 18,000 R packages on CRAN. So a new user trying to figure out how to use R for pharmacometric work, a big question is, which one of these should I invest in learning? And domain specific use cases are probably the best tool for learning. But where to find these? Fortunately, if you work at a big organization, there's probably colleagues you can speak with. There's always internet searches, but the best example is to be able to find an example piece of code to say, okay, this is what I want to do, and then start from that and modify it. From the organizational perspective, there's a desire for harmonized workflows in processes in general. And the reason for this is to simplify staff training and onboarding, to ensure consistency and deliverables to other business functions. This can be a challenge with command line tools. If you have 10 separate users, using an open source tool like R and delivering 10, types of formatted Word documents to the regulatory documentation group, that group is going to be required to do a lot of additional work compared to if they receive the same, format each time. In addition, organizations, they need domain-specific maintenance and support. Very often with the implementation of our solutions in pharma, you have business users that are assuming key roles in maintenance and support. And this certainly is not ideal. These people are scientists first, not support folks. And it's not an ideal situation, but in fact, in a way, it's driven by the complexity of the domain. While at Roche, I was it's interesting, kind of a funny story. While at Roche, I was a user of S+, and I kind of served as the S+ troubleshooter in my clinical pharmacology department. And one day somebody came into my office and said, Keith, I'm having a problem. I looked at the problem, I couldn't solve it. And I said, you have to call IT support. So they left my office. They said, okay, thanks very much. And 10 minutes later, my phone rang and it was IT support. They said, yes, Keith, someone has a problem with their S+. So it went in a big circle. So finding support for these solutions can be an important, issue for organizations, wanting to have sustainable, stable working environments. So the strategy behind RSNLME really is to provide a solution that meets both individual and organizational needs. NLME is a capable analysis engine. PML is a capable modeling language. There are many types of models supported. I provide a link to PML School, which gives a good overview of the types of things that you can do with PML. PML is a shared language between Certara tools. Users can grow into RSNLME from other Certara tools. I mentioned we support key workflow steps with Shiny apps that teach R, and we've designed it to work hand in hand with other R solutions. We have plans to support, you provide expanded workflow support beyond the analysis. So really the work we're doing for report generation is a very good example of that. And in the end, the use of these tools will provide major efficiency gains for the full workflow. From an organizational perspective, RSNLME really adds to the scalable solutions offered by Certara. One can use Phoenix for non-compartmental analysis, which uses the same PML language. So a suite of tools that can support both junior and senior users in the department is of importance. You can combine it with our integral solution, which is a cloud-based data repository with 21 CFR 11 compliance capability. We have a tool called Piranha that I'm not going to discuss, but this is a workbench for management of pharmacometric analysis workflow. And implementation of that at organizations leads to easier workflow harmonization. Stability and quality of the packages, really we're building these like a software development company would build them, following guidelines to ensure quality of our packages. And finally, Certara is able to support these packages to provide support for the use of R in the pharmacometric workflow. And that's important. I mentioned my story at Roche. I wish I could have sent my user to a company that could have helped them better. So I think I have. Am I out of time? Do I have 1 minute for a demo?
You can continue, sir. You can.
Okay, excellent. So what I'm going to demonstrate very quickly, obviously in the time allotted, I can't cover all of the syntax in the tool, but I have our command line code, and I'm going to just show you quickly the concept of the tools that teach the Shiny apps. So here we're working at the R command line. We import a data set. One might do some exploratory data analysis. Here we're using ggplot. Again, Grammar of Graphics is a very popular package in data science. We get a plot. This looks like 2-compartment drug, perhaps. One could expand into other R packages to do further exploratory data analysis. There's a ggquick EDA package. This is an exploratory data analysis package that was developed by one of our consultants in Certara. So we have a long history of actually contributing to open source on our consulting side. That's Samir Muksasi, very talented individual. But for the interest of time, the next step would be to define your PK model. We use, again, this is R speaks NLME syntax. We have a PK model command here. We define a 2-compartment model, and it takes the default for the absorption phase, which in this case is IV is the default, maps the model variables to the data set, and assigns a model name, which will be a directory that contains the model output. So we run that. We then adjust the model, removing random effect terms from 2 of the parameters, CL2 and V2, adding some fixed effects. These are the general steps of setting up a model prior to execution, and we're going to look at some covariates. So we run this and we print the model, and you get a nice summary of the model. And for those of you who know PML, this is exactly the same modeling syntax that is used in Phoenix Win Non-Lin. Now, this looks all very complicated, right? So what happens if you don't know the code? This is where the UIs come in. And the first UI that I will briefly show, and perhaps that's all we'll have time for, is the Model Builder UI. This, again, is an R Shiny app. Everything, the foundation is R. And that's designed, again, so you have the complete flexibility to customize work to your exact problem. And that is the nature of pharmacometric work. What you do next, if you ask any scientist, if you ask them, what are you going to do? How are you going to do this piece of work? And they say, well, it depends. And you ask them, well, it depends on what? Well, it depends. They need to look at the data and interactively decide what to do next. And that's why command line languages are so, so popular. But this interface, just very quickly, there's drop downs. You can very quickly specify the type of model that you are going to fit. Easily over 1,000 combinations are supported. So if you wanted to specify a model, you see the code is immediately updated in this panel here as you make selections. So if I wanted a PK indirect response model with 3 compartments, Michaelis-Menten, elimination, and I want actually a inverse inhibition, you get the idea. The code is immediately updated, and the user does not need to know how to write it. One would step through the next tab, and the tool kind of steps you through building that model. So you would specify covariate influences. Again, I won't do it in the interest of time. Fixed effects, random effects, block statements, that type of thing. And in the end, of course, you can toggle back and forth to your data set. In the end, one can generate the RSNLME code that produces that model. So it's a way that the tool can actually teach user how to write this code. In the interest of time, I think I need to stop there. I hope that gave you a good example of how we've designed these tools, why we think it's important to support R in this domain. And at that point, I'll stop and take any questions.
Yeah. Thanks, Dr. Keith, for a wonderful session for NLME. I request to post Q&A for the participant. Sir, we have one question for: is RS NLMEs are more complex to use when compared to impurity LME?
It's an excellent question. By T NLME, I'm assuming that might be a typo, and you
I think so, sir.
You mean Phoenix NLME?
Yeah.
Yeah, so Phoenix NLME is a graphical user interface tool. The model syntax between the two tools is identical. Where R becomes perhaps a bit more complex is instead of being able to use GUI type tools to work with the models, you're working with a set of R functions. So again, it is slightly more complex. However, what the gain is the efficiencies across the workflow and some of the types of things that I described, as well as the access to the larger open source community and the really bewildering set of tools that are available to assist various elements of the work. And again, that was one of the main reasons for extending the PML language into R. So it is a bit more complex, yes, but the payoff is worth it in my opinion. Yeah.
Okay. Thanks, Dr. Keith. Is any? I think one more question is this. Are you having any session coming up with RS-NLME in India to help us understand its difference and compare it to NLME?
I'm not aware that we have any directly planned, but this is something that certainly could be arranged. I'd be happy to participate in something like that. Yeah, 20 minutes is kind of a short time to dig into a command line language, but yeah.
Okay. Thanks, Dr. Keith, for participating with the Global Virtual Healthcare Summit and sharing your time and knowledge. Thanks very much.
Thank you very much. Have a good day.
Yeah, same to you.